In our cells, DNA is continuously damaged by several factors. Among all types of lesions, the most dangerous ones are double-strand breaks. Double-strand breaks are caused by cellular metabolism products, but also by errors during DNA replication or during special events in the life of some types of cells, such as meiosis. Chemical compounds used in chemotherapy and ionizing radiations of radiotherapy produce a large amount of DNA damage, both in cancer and in normal cells. Since cells fail to repair those huge damages, they undergo programmed cell death. DNA repair mechanisms, as the ones that identify and repair double-strand breaks, in cancer cells do not work properly, as in normal cells, and could become a target for new drugs. This is the case of PARP inhibitors that are used in the treatment of advanced ovarian carcinoma in patients carrying BRCA1 or BRCA2 mutation. BRCA1 and BRCA2 genes are involved in DNA repair. When BRCA1 or BRCA2 are mutated, their corresponding proteins do not work properly or are not produced at all, and DNA lesions accumulate, thus increasing the risk of developing breast and ovarian cancer. In the case of ovarian cancer, despite surgery and chemotherapy, 70% of patients experience a relapse in 3 years. This percentage drops to about 40% if patients are also treated with PARP inhibitors. PARP inhibitors act specifically on cancer cells with mutations in BRCA1 or BRCA2 genes. The cells are not able to repair DNA damage mediated by these proteins and therefore die. Instead, in the other cells of the patient, a copy of the gene works properly, and this is enough to guarantee DNA repair. In June 2019, the European Commission has approved the use of PARP inhibitors in maintenance treatment in cases of advanced ovarian cancer with BRCA1 and BRCA2 mutations. This is an example of how basic research in DNA repair can serve to develop an extra weapon against cancer.